Hello students. This video is regarding a chapter from class 12, and that is microbes in human welfare. Microbes in human welfare. Now this is one of those chapters from where, especially during your competitive exams, you get a lot of questions. So when we speak about microbes, as you can see on my screen, it's already shown that when we speak about microbes, what do we mean by a microbe? It's written over here, microbes. So what does this mean? Microbe means a microorganism. Why is it microorganism? Because we cannot see it with our bare eyes. We need a microscope to see a microbe. Now, in our biological systems, whatever biological systems are there on the planet, the most abundant group of organisms, if I ask you, name the most abundant group of organisms, then readily the answer from your side, it will come that producers, which means green plants or autotrophs, which is absolutely correct. But there is another group of organisms which also form considerable amount, or they occupy considerable position in the biological systems, and those organisms are microbes. Microbes, they can vary. There are various kinds of microbes. They can be bacteria, they can be fungi, they can be viruses, prions. Now what are prions? I hope you remember in class 11 you had got an idea, a very small idea about prions. Prions are infectious proteinaceous agents. Those are called prions. These are mostly proteins and they are infectious. So bacteria, viruses, fungi, prions, these are mostly the microbes, the microorganisms that we are aware about and that we study about. Now, whenever we talk about microorganisms, especially in these times, these are the times of pandemics when the novel coronavirus, it's changing its form each and every month or 2 or 3 months alternatively. It's mutating so rapidly. So we always think about the harmful aspect of the microbes. Whenever we think about microbes, the first thing that comes to our mind is, okay, so this microbe causes this disease, tuberculosis caused by this bacteria, cholera caused by this bacteria, coronavirus, this COVID is caused by a virus. Okay, and there are numerous other communicable diseases which are caused by microbes. But we should know that microbes can be put to some good use also. They can be very helpful in various aspects of our life. In various aspects. Like, as my screen tells you, a very important usage of microbe we can find in our household, in our own kitchen. You have often seen your mothers preparing curd, dahi, right? And the chach, the yogurt. Yogurt and dahi more or less same. Chach, what is chach? Any idea what is chach? Chach is, in English we can call it buttermilk. Okay, then the famous cheese, which we often use in our kitchen for making various dishes. Now for all these items, food items that I mentioned, without microbes, their manufacturing is literally impossible. So microbes have a very important role in our household, in our kitchen. Okay, Then microbes are very useful in the industry also. They have a wide range of industrial application. We will go into that. And biogas, which is very much in discussion now, because biogas is an alternative. It is thought to be an alternative fuel now. We are trying to depend less on the fossil fuels and more on these biogases, because number one, these are eco-friendly sources of fuel. The world is ready to move towards methods, towards procedures, towards certain designs which are eco-friendly in nature. So biogas is an eco-friendly alternative to the conventional sources of energy that we are we use for so many years. And then comes this one, STP. Now what is STP? STP means sewage treatment plants. Sewage treatment plants. Now what is that? We'll go into the details. So we'll first start with use of microbes in household. So let us go back to the slide that I was showing you. See, what is this slide telling you? That in our kitchen we are using microbes for preparing some day-to-day food items like curd, yogurt, buttermilk. These are very much in use. We almost consume them daily. Now microbes, the microbe that is used in preparing curd, in preparing yogurt, they are called lab. They are actually called lab. If I write down, they are called lab. What is this lab? Lab means, lab is nothing but lactobacillus. Lactobacillus or lactic acid bacteria. This is called lab. This is the full form of lab. Now, under this lactic acid bacteria, there can be many examples. These bacteria, they basically ferment the milk. They ferment the lactose of milk and converts the lactose to lactic acid. So they are called lactic acid bacteria or lab. Very, very important. One of the examples of lab is, as my screen tells you, Lactobacillus acidophilus. Lactobacillus acidophilus. That is a very important example. Another example is, you may refer to Lactobacillus bulgaricus. Okay, that is also a very important lab, an example of lab. Okay. Next comes another important food item where there is extensive use, and that is idli dosa. Idli and dosa, these are basically fermented products. What is being done? What is the composition of idli dosa? In case of idli dosa, rice flour is taken. You know what is rice flour? We know what is flour. And whenever we talk about flour, we think about wheat. But rice flour is obtained from rice, from paddy basically. So rice flour and gram flour or gram, if they are crushed and made into a paste, into a batter, then that batter can be acted upon by certain microbes which will ferment the starch that is present in the carbohydrate and will produce ethanol, which is present, and carbon dioxide, which is present in idli dosa. How does a idli look like? Idli looks like as if it has got a puffy and a porous appearance. Puffy because when the bacteria, they act on the batter which is formed, the rice flour and the gram, then what happens? They ferment the starch present in the batter. First, the starch is broken down to glucose. Then glucose, it undergoes further oxidation and it forms pyruvic acid. Pyruvic acid then is converted to ethanol and CO2. So let me tell you what is done over here. Let me write down. So I can write over here, starch becomes glucose, becomes pyruvic acid, and this pyruvic acid, it is further digested and converted into ethanol. And this ethanol actually gives that sour taste. Not only ethanol, also carbon dioxide, CO2. As CO2 accumulates, the batter swells up. Now when it is baked, due to the high temperature, CO2 comes out, CO2 evolves, leaving behind the pores. That is why idli, dosa, they have a porous appearance. Those holes are made by CO2. CO2 comes out and makes the holes. Okay? And this entire process that I told you, from starch to glucose to pyruvic acid to ethanol to CO2, this is possible only anaerobically. So this is anaerobic digestion. But some enzymes are required which can be secreted by these microorganisms. Like in this case, streptococcus is a very important microorganism for idli dosa. So streptococcus basically does this because it secretes certain enzymes. Those enzymes facilitate this entire fermentation process. So ultimately what is formed? Ethanol is formed and carbon dioxide. These two things are formed. Okay? Coming to cheese. As you can see, the next item that we will be talking about is cheese. A very, mouth-watering delicacy for many people across the globe. It has got widespread uses. We can use it on breads. There are many preparations that are done with cheese, right? Now cheese, there are various kinds of cheese. One of the most popular forms is Swiss cheese. Let me write down. Swiss cheese. And can you see that on top of it I have mentioned the name of the microbe, Propionibacterium. So Propionibacterium is the name of the bacterium which is actually used in preparing this Swiss cheese. And Swiss cheese, what is the speciality? If you have seen Swiss cheese, it has got large holes on it. Large holes on it. Why large holes? Due to the presence of, or due to evolution of, carbon dioxide. Carbon dioxide evolves and those holes are made. Not only Swiss cheese, there can be other kinds of cheese also. Roquefort cheese. Okay? And then we have camembert cheese. All these kinds of cheese, they require different kinds of microbes. Most popular is Swiss cheese, and the microbe that is used is Propionibacterium shermani. Propionibacterium shermani. That is used in making Swiss cheese. Next comes bread. Above bread, what have I written? I have written Saccharomyces, which is basically nothing but yeast. So Saccharomyces or yeast is a very useful microbe that is being used for making bread. That's why it is also called baker's yeast. Please remember the name baker's yeast, Saccharomyces cerevisiae, baker's yeast. What it does the same thing. It converts starch to glucose, glucose to pyruvic acid, pyruvic acid to ethanol. So the same process it does, fermentation. Now during this process what happens? How is a bread prepared? We need wheat flour this time, not rice flour. Wheat flour, which we call maida. So wheat flour, and we take water, we add some salt and sachets of this yeast powder. These are available in the market. So this yeast powder, these are basically stirred in lukewarm water so that the yeast cells get active. Dough is prepared. The wheat flour, water, salt, these are mixed. A dough is prepared. Then this yeast cells, these yeast cells are added to the dough. Yeast cells were already activated because they were added to lukewarm water, so that activates the yeast activity, yeast cell activity. Yeast can secrete different kinds of enzymes. One of the most important ones is zymase. So let me write down over here. One of the most important is zymase, but not only zymase, it can also secrete two more. One is amylase and the other one is maltase. Zymase, amylase, maltase. So these are the enzymes which act on the dough. As a result, what happens? Ethanol is produced and carbon dioxide is produced. Now when carbon dioxide is produced, it causes rising up of the dough. The dough swells up, and that process is called leavening. Remember the term, it's important. The process is called leavening, swelling up of the dough due to accumulation of carbon dioxide. That is called leavening. Now when this swollen up dough, when it is baked, when it is subjected to high temperature, what happens? Carbon dioxide escapes, leaving behind pores, leaving behind holes in the dough. And that is why bread is porous, bread is fluffy, bread is lightweight. So remember the name of the microbe, which is Saccharomyces cerevisiae, which is also called baker's yeast. Remember the names of the enzymes: zymase, amylase, maltase. Zymase is very important. Okay, all of them, they help in which process? They help in fermentation. So this is important. This comes in the MCQs also. Okay, now very popular drink in southern part of India, South India, is, as I have written over here, you see, toddy or toddy, you may say toddy. Different pronunciations are there. So this one, how is it prepared? It is basically prepared by fermenting palm sap of the palm fruit. It is fermented and then this is prepared. So you can understand that this is another application of microbe. Okay. Now this toddy, it is prepared by this, using this microbe, Caryota. This is the name of the microbe, Caryota. Now another important food item very popular in Chinese dishes is soya sauce. We all know that. Now how come soya sauce is formed? Soya sauce is formed by making, or it is prepared from, as you can see on the slide, it's written soya bean and wheat. Soya bean and wheat, these two are mixed, then they are fermented. After fermentation, the dark brown sauce that is obtained, that is received, is soya sauce, and the food that is left behind becomes tofu. Tofu is very, nowadays it's very common here also. Tofu is very much, in Western countries, we use paneer over here. So they use tofu like paneer. But in case of paneer, it's a dairy product. Tofu, it's a soya product. Okay. So after fermentation, the food part, the solid part that is left is basically tofu. And the liquid part, the dark brown sauce, that is actually soya sauce. So while we are learning household products, use of microbes in household products, we will have to go through these products that are there on the slide. We have to remember their names of the microbes, and if any enzymes that are there. So this is all about household products. Now next comes industrial products. Very, very important application of microbe in industry is fermented beverages. Okay, beverage. And by beverage we mean wine, we mean beer, then comes whiskey, brandy, rum. These are the fermented beverages that are industrially prepared, and it's a huge market, we all know. It is industrially prepared on a large scale, and for that, industries, they deploy large fermenters. I'll show you one picture. See, this is one picture of brewery fermenter. So this is one picture where a fermented beverage, this is a factory basically, and these are the fermenters where the process is done. Okay, so this is a brewery fermenter. So what is being done now, depending on what kind of beverage we want to prepare. Suppose if we are trying to prepare wine, okay, it's written over here molasses. Now what is molasses? Generally molasses are obtained from the fruit juices. The fruit juices, from there we get the molasses. So wine is prepared from fruit juice, the grapes generally. And then comes the beer. Beer comes from malt. So I'll write over here. Wine, we can get it from the grapes. Beer, we can get it from malt, barley. Okay. Then comes whiskey, brandy and rum. Now let me tell you, molasses is a very good source of rum. Brandy, we can get brandy from fruit juices. And whiskey, we can get it from the fermented cereals. So the malted cereals. I have already written the sources. Please see carefully. Grapes, from grapes we are getting wine. From barley we are getting beer. From cereals we are getting whiskey. From molasses we are getting rum. From fruit juices we are getting brandy. Now see the slide carefully. Wine and beer, these two beverages, the alcoholic percentage is very low. Especially in beer and wine. In beer, the alcoholic percentage will be hardly 3 to 6%. Okay, but in whiskey, brandy and rum, especially in brandy contains the highest percentage of alcohol. Highest percentage of alcohol is found in brandy. It is almost 60 to 70% brandy contains. Okay, now it depends on whether distillation has been done or not. Wine and beer are such beverages which doesn't require distillation. Okay, and whiskey, brandy and rum, they require distillation. So the more the distillation process takes place, the percentage of alcohol or the alcoholic content increases. Lesser the distillation, alcoholic percentage decreases. So this is a very important difference, students. Keep it in mind. Wine, beer, no distillation. Whiskey, brandy, rum, vodka, all these popular beverages, they require distillation and their alcoholic percentage is much more. Highest is in brandy, almost very low, or we can consider lowest in this beer. Okay, so this is all about fermented beverage. Now next comes antibiotics. Who discovered antibiotics? We all know Alexander Fleming, and there is a story behind that. So he was basically culturing staphylococcus bacteria in his lab. So after a week of his culture, he saw that half of the bacterial colony has turned white. Then he discovered that a certain kind of a mold has developed in that bacterial colony. And then he discovered the mold to be, it's written over here, Penicillium notatum. The mold was nothing but it was Penicillium notatum. So Penicillium notatum was secreting a chemical, secreting something which was killing the bacteria. So from there the concept of antibiotics came. So what are antibiotics? Antibiotics are chemicals that are produced by certain microorganisms which kill other microorganisms, but they will only kill the harmful microorganisms. If any antibiotic kills a useful microorganism, that is not a good antibiotic. We consume antibiotics, but doctors always tell us indiscriminate usage of antibiotics is deadly. Because when our body gets, used to or accustomed to antibiotics, it destroys the natural flora of our intestine. Natural flora of our intestine gets hampered. Flora means our gut has got certain bacteria. One of the most important is E. coli, Escherichia coli. More we get dependent on antibiotics, those antibiotics will have an effect on E. coli, on the useful flora of our gut. So that will be deadly for us because we need E. coli for digesting the food. We need E. coli for synthesis of vitamin in our body. Okay, so now Alexander Fleming was the scientist to discover antibiotics. Who named it? Who coined the term antibiotics important? So it will be, who coined the term antibiotic? Salesman. Okay, Selman, he coined the term. So I'll just write it down. Okay, one more thing. I'll write 3 names over here. Okay, one is, let me write Alexander Fleming. Okay, then I'll write Ernst Train, and then Howard Florey. Howard Florey. Now, why have I written these 3 names? They 3 together got the Nobel Prize for discovery of antibiotics because Alexander Fleming only observed the phenomenon, he discovered, but the usefulness of antibiotics, that was described by Ernst Chain and Howard Florey, and that's why their names are also important. We should remember all the three names. It's important for our boards as well as for our competitors. Okay, so we need to remember this. Okay, and they got the Nobel Prize in 1945. Okay, now antibiotics, I told you that when can we consider them to be a good antibiotic? When they have they do not affect our gut flora, they keep our gut flora intact. We are not having any problem with the flora. And they act on a wide range of microorganisms. So a wide spectrum antibiotic is always preferred. One of the first, oldest antibiotics discovered was penicillin. As you can see on the slide, there are more examples of antibiotics. Now, antibiotics, more examples if you want. The most important is penicillin, but other antibiotics are also there, like, I'll write down over here, chlorotetracycline. And then you have this one, erythromycin. Remember these two names because these two are popular antibiotics. Chlorotetracycline, erythromycin, and the most popular antibiotic now is azithromycin. We all know, those who have suffered from this novel coronavirus attack, they were prescribed azithromycin, a very useful, potent antibiotic. So apart from penicillin, we are going to remember these names also. I'm writing down azithromycin. Okay, so this is all about antibiotics. Now next comes bioactive molecules. Now, what are these bioactive molecules? Bioactive molecules are certain compounds, certain chemicals that are useful in our body in some way or the other. Now these compounds, they can be produced inside the body, okay, or they can be added from outside, but they are functional in a living system. They are functional in a living system, or they can interact with the components of living system. Very important. Bioactive molecules are those molecules which are functional in a living system, or when they get inside a living system, can interact with different components of the living system. Under this, we are going to learn these examples that I have already written. Organic acids. Organic acids are important. Certain microbes are there which can convert carbohydrates to organic acids. Okay, like citric acid, very important. Why citric acid is important? We are talking about industry. Citric acid must be having industrial application. What is that application? Citric acid is employed in dyeing, it is employed in coloring. Okay, then comes it is used in preparation of medicines. Citric acid. Then flavoring of food. These are the uses of citric acid. Another important is acetic acid. Acetic acid is used for preparing vinegar. Very important. Butyric acid, it is used for making rancid butter. Then lactic acid used in curd. Please learn these examples. Okay, so citric acid, acetic acid, butyric acid, lactic acid. These are the examples of organic acids you are going to learn. Learn their industrial application. Citric acid used in inks, in medicines, in dyes. Lactic acid used in curd. Acetic acid used in vinegar. Butyric acid used in making rancid butter. So these are the applications, students, remember. Then comes alcohol. Just now we have discovered about the brewery industry, the beverage industry, how microbes are useful in making alcohols. Then comes enzymes. Enzymes like, I'll write down over here. Learn these names. Lipase. What is lipase used for? Lipases are used in detergents. And these lipases, basically, they are responsible for removing those greasy stains on our clothes or on anywhere. Lipases are present in detergents, and lipases, they are helpful in removing greasy, oily stains. This is the use. Learn the names of the microorganisms. Candidolipolytica. Candidolipolytica. And another one is Geotrichum candidum. So two microbes we are going to learn which can secrete lipase. One is Candidolipolytica. Another one is Geotrichum candidum. These two. Okay. Next comes pectinases. The fruit juices that we buy from market. When we eat the fruit, when we drink the fruit juices, do we find anything gritty? Do we find any pulp of fruit? No. We actually drink a clean, clear fruit juice, and that is why they are also not much prescribed by the nutritionists and the doctors also, because they say these marketed fruit juices are low in fiber. Why? Because when these fruit juices, they are prepared industrially, then pectinases and proteinases, I'll write down the names. Pectinase and proteinase. These enzymes are used. What are they used for? Pectinase is generally used to degrade pectin. Pectin is an organic substance which binds the fibers in a fruit. In any fibrous plant, in any fibrous part of a plant, pectin is the compound which binds the fibers. So pectinase will degrade that pectin. As a result, what happens? The fiber content goes down, and that is why the marketed fruit juices are low in fiber content. Proteinases also. Proteinases, they have the same use. They basically clear the fruit juices. Learn at least two names of microbes from here. Very easy one will be to go for bacillus. Bacillus can secrete protease. Okay. Aspergillus, protease. Then pectinases are secreted by Aspergillus niger. Aspergillus niger. And proteases are secreted by another species of Aspergillus and Bacillus, and also Mortierella renispora. Mortierella renispora. Mortierella renispora. These are the microbes. What did I say? One is Aspergillus niger, which secretes pectinase. Proteases are secreted by Aspergillus, maybe some other species, and Bacillus. Okay, and Mortierella renispora. So these three, they secrete protease. Pectinases, proteases clarify fruit juices. These are the industrial applications. Another one is, as you can see, it's written cyclosporin A and statins. Now, what are these? Cyclosporin, cyclosporins or these statin kind of compounds, they are basically those which in medical terms we call them clot busters. Why? Because if a person suffers from cholesterol deposition in his arteries, that person will be prone to heart attack. Now, when a person suffers from heart attack, what happens is the person is taken to the hospital, and immediately the person is administered with an enzyme which is called streptokinase. Streptokinase is a clot buster which will clear the cholesterol deposition so that the artery gets it gets cleaned and blood flow can be restored. This comes in the competitive exam, this question. When a person suffers from heart attack, myocardial infarction, when the person is taken to the hospital, then which medicine is given? So there can be different brand names, but the composition is streptokinase is given, which is a clot buster. Many people regularly take doses of statins because that is good for their heart health. Okay, they take statins. Cyclosporins, they basically, how do they work? Cyclosporins competitively inhibit the enzymes that synthesize or that catalyze the synthesis of cholesterol in our body. Cyclosporins inhibit those enzymes which are the catalysts for cholesterol synthesis in our body. So basically, cyclosporins are inhibiting further cholesterol synthesis. And as a result, cholesterol deposition is also restricted. So this is the use of cyclosporins and statins. They are called clot buster for removing the clots from the blood vessels of patients who have undergone myocardial infarction. In easier terms, we call it heart attack. Okay, so these are the examples of bioactive molecules. Okay, there are many more applications of microbes, students. So we are going to go through all of them. But for today, I think we have discussed about the lab, lactic acid bacteria, usage in our household, usage in the industry. These two aspects we have done, we have covered. Under household, in which food items, which microbe is used, which enzyme they secrete, we have learnt. Under industrial products, beverages, antibiotics, and these bioactive molecules, these three are the most important ones. We need to go through them through the names of the scientists, names of the beverages, their sources. We must remember beer contains lowest alcohol and brandy highest, from where they are derived, which is produced by distillation, which is not produced by distillation. This we need to remember. So if we can remember these things, we are ready to prepare for our exam, because this chapter is basically an easy chapter, students. What you have to do, you have to do a mugging up, because you have to memorize the names of the microbes, the products, and the enzymes. But if you give it a serious thought, it's a very, scoring chapter, right? So if questions come from this chapter, you should definitely go for it, because this only requires, memorizing. Okay, so household products, industrial products. In the next video, we are going to discuss about some more usefulness of microbes. So till then, bye bye. See you in the next video.